So, now let us go to the detailed part of each of the processes involved in studying two-dimensional electrophoresis, the experimental workflow. There are different steps which are involved in making a good gel. So, the first point in that workflow is isoelectric focusing, which is the first dimension separation of the proteins based on isoelectric point. Second step is equilibration of immobilized pH gradient strip. When you are running an SDSPAGE gel, you make your protein denatured, you want to separate that based on the molecular weight, and you heat the samples, you apply different type of denaturant. Now, in this case, when you are doing 2DE, your first dimension separation has occurred on the IEF in the immobilized pH gradient strip. Now, you want to take these strips and want to apply that on an SDSPAGE gel. You cannot do the heat treatment. So, you want to make these strips prepared to be separated based on the molecular weight in the second dimension, and that is where the equilibration process is useful. Once you have done the equilibration of these strips, then protein can be further separated based on the SDSPAGE, the third point. Now, fourth in the workflow is staining the gels and visualizing the gel images after staining with different type of stains. Image analysis, the fifth point, is very crucial because you need to analyze all the protein spots so that the abundance of each of these proteins can be calculated and more detailed statistical analysis can be performed. Once you are confident that these are the proteins which are highly significant, then you would like to pick those spots by a process mentioned in 6th point, which is spot picking. After that, if you want to identify and know that what this protein is, then you need to do the in-gel digestion or enzymatic digestion of these proteins obtained from the gels. Then the last point will be the mass spectrometry analysis, which will be different series of lectures in the other module based on the mass spectrometry. So, I will now show you an animation for two-dimensional electrophoresis, which will give you an overview of different steps involved in 2D experiment. So, in this animation, I will describe you the two-dimensional electrophoresis process. Prior to isoelectric focusing in two-dimensional electrophoresis, the commercially available IPG strips must be rehydrated. This process can be done either by the passive rehydration or active rehydration. In passive rehydration, the IPG strip is placed with its gel site downwards in a well containing the protein sample reconstituted with a suitable buffer. As you can see in the animation, you can apply the protein sample and then add the IPG strip so that it can absorb the protein solution. This process can be done for 10 to 20 hours depending upon your length of the IPG strip. This is then covered with mineral oil to prevent the gel from drying up and left overnight. Other process known as active rehydration, where the protein sample is added to the strip via a sample cup followed by the cover fluid to prevent the gel from drying up. The protein sample being applied, a very low voltage condition is provided and then cover fluid can be added. This is then placed in the isoelectric focusing instrument and low voltage is applied. Process can be performed from 10 to 20 hours. These loaded strips are then focused on an isoelectric focusing unit by passing the current. The various proteins of the sample mixture migrate in the electric field and come to rest when the pH is equal to their isoelectric point or Pi. So, they become neutral and are no longer being affected by the electric field. The progress of electrophoresis is monitored by adding a tracking die, which you can see in the animation moving ahead of the proteins. The IPG strip is then equilibrated in a reducing agent such as DTT, followed by an alkylating agent iodoestramide, which prevents reformation of reduced bonds. This strip containing the separated proteins is then placed on SDSPAGE gel slab for further protein separation in the second dimension based on the molecular weight. The proteins on the IPG strip are subjected to SDSPAGE by applying a direct current between 100 to 350 volt depending upon the size of the gel. Any protein that may have been present as a single band on the IPG strip, as you can see in the red circle, due to similar isoelectric point, can then be separated based on the molecular weight, where the smaller proteins will migrate the farthest. This is view of a sample gel which has been run by two-dimensional electrophoresis and stained with Kumasi blue. Each spots provide the information about the molecular weight and isoelectric point of the protein. So, let us talk about isoelectric focusing or IEF. In the IEF process, proteins are separated according to the isoelectric point. You can take the immobilized pH gradient strip and apply the protein solution on the IPG strip, so that protein is absorbed on the strip, and then after applying the electric field, the proteins will migrate according to its charge. When protein is reaching to its isoelectric point, where pH becomes equal to its isoelectric point or Pi, then proteins do not move in the electric field because of the lack of charge. So, when pH equals to Pi, then protein will not move any further. Let me show you this process in this animation here. You have an IPG strip with pH 3 to 7 gradient. When you are moving in the electric field from the positive charge towards the negative charge, when pH becomes equal to its Pi, then there is no net charge and protein will remain over there. So, now this will be the isoelectric point. Now, in this view, you can see from the low pH to the high pH, proteins are moving in the electric field, and once they reach to a stage where there is no net charge or pH becomes equals to their isoelectric point Pi, then these discrete band can be seen. So, the rehydration is one of the important steps before applying the electric field in the IEF process. As you have seen in the previous animation, that to rehydrate the IPG strip, you can apply the protein solution on the IPG strips, and different methods can be used for this, whether passive rehydration where no voltage is applied, or active rehydration where you apply the protein sample and a very low voltage is applied to resolve the proteins. Now, we have already discussed that none of these methods can be compared and said as a superior method. One has to really use that in their own biological sample and see where the protein separation can be better based on whether passive rehydration or active rehydration. Once you have done the rehydration process, then one can apply the mineral oil to avoid any protein evaporation from these IPG strips. Now, different type of strips can be used during the IF process, whether its pH gradient of 4 to 7 or 3 to 10 or some other pH range. As I described in my previous slide, depending on your biological question, if you are mainly interested in separating the proteins in their biological pH range, then 4 to 7 could be a good choice, where you can have better separation of the protein, but you may lose few proteins which could be in the extreme region of acidic and basic. Now, if you want to know all the proteins which could be present in your sample, you can apply a very broad range strip like 3 to 10. Now, what type of length of IPG strip should be used? One can use starting from 7 centimeter till 24 centimeter. There are different type of strips being manufactured from different commercial companies. Now, when you are optimizing a protein extraction protocol, it is better if you use a small strip so that you know that your IF process is going fine and your protocol is looking good. You can separate the proteins. Now, once you know that your extract is good, then you can apply that to separate the proteins in the long strip length, for example 17 or 24 centimeter. Now, these IF units or isoelectric focusing units obtained from different vendors, commercial manufactured, they are capable of taking the IPG strips of different varying pH length. Few instruments take a flexible length from 7 to 24 centimeter. Other commercial instruments, they are able to take different type of trays designed for each type of strip length. Now, depending on your proteins, how much protein sample you have in your protein in the sample mixture, it's always better or good idea to resolve that on the very large gels. For example, 18 or 24 should be a better choice. But if you do not have enough protein to separate, then you have to restrict yourself to smaller strips. Or one can still go with a large strip but apply different more sensitive strain. So, when we will talk about different type of staining methods, at that time we will talk, even if you have low protein, you can separate on the large gels, but then apply more sensitive stain such as silver or cyproruby. But overall, the large gels are recommended for doing any type of differential proteomic analysis when you want to compare your controls and treatment spot. But handling the large gel is very tedious. So, I will show you some lab demonstration, the video for that, so that you can be well prepared to perform these experiments in your lab. These are some recommendations for how much protein one should load on the IPG strips. For example, if you have a small strip like 7 centimeter length, you can apply between 100 to 300 microgram of protein, and maximum volume could be 150 to 160 microliters. Usually, Kumasi stain is a good choice, and if you have Kumasi stain, you can stain that with this much protein. If your protein sample is having very less amount of protein, for example 10 to 100 microgram, at that time you have to apply the sensitive stains such as silver or cypro-ruby. One can also apply the large strip like 17 centimeter or 24 centimeter strip, but then you have to increase the protein amount to be loaded and separated on the IPG strip. For example, between 250 to 1000 microgram of protein can be loaded on the large strip, and one can visualize that by using Kumasi stain. Total reaction volume should not be more than 350 microliters. But if you do not have too much protein present in your sample, then still you can apply this method. You can apply the large strip, but then you have to stain with silver or cypro-ruby. In that case, you can load between 100 to 100 to 1000 microgram of protein. So, I will give you the laboratory demonstration of isoelectric focusing, so that you can be very clear about different intricate steps involved in performing IEF.
Isoelectric focusing. This process involves two major steps: rehydration of the IPG strips and focusing of the rehydrated strips. IPG strip rehydration. Clean all the apparatus thoroughly before use to avoid any contamination. Add the reconstituted protein sample in a well of the rehydration tray. Then remove the IPG strip from its cover and place it carefully in the well. IPG strips are used for separation of proteins based on their isoelectric point. These are acrylamide coated plastic strips containing immobilins of various pH spread across it. They have successfully replaced conventional tube gels due to their reproducibility they can achieve. After around 30 minutes, pour some mineral oil over the strip to prevent it from drying up. Cover the tray and leave it overnight to allow rehydration to occur. Isoelectric focusing. Initialize the instrument and clean the surface well with a dust-free cloth. Place the focusing tray on the instrument and ensure that it is properly balanced. Cut the paper wicks required for focusing to a suitable length and wet them with a small amount of water before use. Carefully remove the IPG strip from the rehydration tray and drain out any excess oil by blotting it on a tissue paper. Place the strip in the focusing tray and immerse it with mineral oil. Then place the two wicks at either end of the IPG strip, followed by an electrode at each end. Fill all the adjacent wells with mineral oil to ensure uniform current flow. Then input the desired protocol on the instrument software along with details of strip length, pH range and number of strips and start the focusing process. The voltage time curve will appear based on the protocol that has been set. Protein separation occurs on the basis of net charge of the protein. Proteins will migrate along the strip and come to rest at a point when their net charge becomes zero, known as their isoelectric point.
Once you have performed the isoelectric focusing or IEF, you need to prepare your IPG strip for the next step, which is SDSPAGE. When you are doing the conventional SDSPAGE, you pour the gel, both resolving and stacking, and then you want to make some wells by using a comb, and then you load your protein samples in different wells. Now, when we talk about two-dimensional electrophoresis, it is different than only doing the SDSPAGE alone. The concept of SDSPAGE will be involved, which will be same, but here there will be variation. You do not need to add the staking gel. You need to prepare your protein sample, which is already focused inside the strip in the first dimension. Now, this IPG strip, which was already put in the electrophoretic unit in the IEF instrument, after that you want to prepare that so that protein can be separated further based on the molecular weight. Now, you need to add SDS to provide the negative charge, and you need to do some treatment for reduction and alkylation. When you are taking the protein sample alone, you have flexibility of boiling the protein sample in the tubes and do all those treatments to denature the protein separately. But now, your protein is already focused inside the IPG strip. So, you do not have that flexibility here. So, all you need to prepare your IPG strip in such a way that you can apply that on top of a SDSPAGE gel. So, I will show you what steps are required in the equilibration step. So, after the first dimension separation is done, you need to coat the protein with SDSPAGE for making it compatible for SDSPAGE. So, you need to coat with SDS, sodium dodecyl sulfate, which is an anionic detergent, and it provides negative charge later on for separation in the electrophoretic field. The equilibration step performs cleavage of both inter and intra chains of disulfide bonds, and it alkylates the sulfhydryl groups of the cysteine residues. So, there are two equilibration steps which are required for this preparatory step. The first equilibration step and the second equilibration step. Your recipe for equilibration buffer is ideally similar in both first and second equilibration, which includes 6 molar of urea, 2% of SDS, 0.375 molar of tris HCl, pH 8.8, 20% of glycerol, and then you need to make one variation in this buffer composition. Either you add DTT, dithiothriotol, or you add IAA, iodoacetamide. Please note, this recipe is given only for your reference. People try different slight modification of this concentration and recipe for doing the equilibration step. Now, as I mentioned, in this buffer you can add DTT, and that will be used in first equilibration step. The first equilibration step can be performed for 10 or 15 minutes depending upon your strip length. Now, once you have done the first equilibration, you need to remove that solution and add a new buffer for second equilibration. Now, this buffer recipe is also exactly similar to the last one, except one change: DTT is replaced with iodoacetamide. So, you can add 135 millimolar of iodoacetamide during the second equilibration step. Now, this is very important because IAA can alkylate the residual DTT and it minimizes the vertical streaking. It also prevents the reoxidation. So, you want to really ensure that your protein is denatured again after doing the IEF process. It is coated with SDS molecule, so that now you can separate the proteins in the second dimension based on molecular weight. This step is also performed for 10 to 15 minutes depending upon the strip length, and once both the steps are done, then you are ready to perform the SDSPAGE. I will show you in this slide the brief stepwise procedure how you can do the first and second equilibration. So, first strip can be equilibrated in the first equilibration buffer by adding inside a tray in a well, and then you change that with the second equilibration solution. The DTT reductant will be used for the first step, followed by IAA, iodoacetamide, which functions as a scavenger of the excess reductant and alkylates and prevent the reoxidation. So, here you can see how different bonds are broken from the disulfide, and IAA is preventing the reoxidation, alkylating these residues.
Place the thawed IPG strip in the tray, and over it pour the first equilibration solution consisting of urea, tris HCl of pH 8.8, glycerol, SDS, bromophenol blue along with dithiothriotol or DTT. Place the assembly on a mechanical shaker for 10 minutes. The dithiothriotol enables cleavage of all disulfide bonds present in the proteins on the IPG strip. Next, add the second equilibration solution to another well of the tray. This solution has the same composition as the first, except that the DTT is replaced by iodoacetamide. Transfer the IPG strip into this solution after draining off any excess liquid from the first. Again, place the assembly on the shaker for 10 minutes. Iodoacetamide prevents reformation of the disulfide bonds by binding to the free sulfhydryl groups that were obtained after DTT treatment.
I hope now you are able to follow the workflow of 2D and will continue in the next class. Thank you.